Hello again and welcome to Chapter 11. This chapter covers autonomic function and a little bit of somatic motor control. We'll include the motor fibers, but the goal here is to help you understand the mostly subconscious and self-regulating aspect of the central nervous system that changes heart rate and breathing rate and other basic functions for survival and homeostasis, and the neurotransmitters that are involved. One of the reasons why I am so into teaching this chapter in this course is that among the most common adverse drug responses are autonomic in nature. That is to say, vomiting and nausea, constipation, perhaps maybe a little bit of drowsiness in some cases, diarrhea, that sort of thing. Those are all somewhat or directly related to autonomic function. And I'm focusing on adverse drug responses here, like I said, these ones here, but it doesn't have to necessarily be in a living patient. If a patient has autonomic issues, or we're taking something that had some side effects or manifestations in the gastrointestinal or other areas of the body that are under autonomic control, that may lead you in the direction of cause of death or those sorts of things. So it doesn't necessarily have to be in medicine. It may also be in things like forensics. So we have two major divisions of the central nervous system. We certainly have the somatic motor division, which is mostly tasked with what we call voluntary type functions. And then we have autonomics, which is primarily involuntary, subconscious, heart rate, stomach and gastrointestinal, what we call tone, how often they're contracting, secretion of glands and so forth, involuntary responses, blood vessel dilation and so forth. Somatic, on the other hand, like I said, voluntary. We're talking about skeletal muscle primarily, though there are some skeletal muscle organs that are under autonomic control, and there are some ways of training yourself to voluntarily alter the autonomic nervous system. The major anatomical difference in terms of the structure of this neural pathway between somatic motor and autonomic is how many neurons are involved. So these little circles right here, that little circle, that's a cell body, and then here's the axon going out in the presynaptic terminals out here interfacing with this muscle. It is one neuron from the central nervous system to the skeletal muscle or the somatic organ of choice. With autonomic, it's a two-neuron system or a two-step system. If we're talking about the adrenal the adrenal glands, like epinephrine and norepinephrine from the adrenal gland. In any case, here's that first neuron. We call that a pre-ganglionic neuron. That's because the synapse for that first neuron onto a second neuron, or in the case of the adrenal gland, a second step, is in what we call a ganglion. So it's pre-ganglionic and then post-ganglionic. That's a very important term to have and to know, because you're going to use that when we're describing the neurotransmitters that are involved. So we have a two-step process, mostly two neurons. But in the case of the adrenal gland, we have a two-phase signaling pathway. And that is part and parcel of the sympathetic and parasympathetic systems. And we're gonna articulate not only the neurotransmitters involved, but we're going to give you some shortcuts to identify one versus the other. So when you think autonomic nervous system, think autonomy. You're thinking things that are generally outside of cerebral conscious control, smooth muscle, heart, glands, and even adipose tissue indirectly through the endocrine system. Mostly involuntary, though it is possible to make them more voluntary, either with training or occasionally you'll have somebody who has a unique ability to do certain things, almost like a superhero kind of ability, but that's pretty unusual. You have two major divisions, as we mentioned. We have sympathetic, which is about mobilizing energy and releasing energy to meet increased need for increased metabolic need or potential for injury or stress. Parasympathetic, on the other hand, is about storing energy. It's about taking external energy that's in the bloodstream and converting it into stored energy for times of need later on, and low metabolic requiring behaviors, if you will. It is intimately associated with the endocrine system, and that's where you can have influence on these not only through neurons like you can in the cardiac, but also endocrine for cardiac and adipose. Behavioral state systems, that's just a fancy way of saying the complex behaviors that we often study in psychology. Psychology, in my mind anyway, is studying the manifestations of behavioral state systems. So the neuronal circuitry is triggering a behavioral state that then triggers a complex behavioral response. All for the sake of maintaining homeostasis, either in the need of stress or injury, or in times where you have excess energy and it's time to store it. Brain structures, we're not talking about the cerebrum, we're talking about the lower brain, if you will, the pons and the medulla and hypothalamus, those types of structures. And just a nice figure from your book showing the sort of oppositional would be the phrase, oppositional effects of sympathetic and parasympathetic. But I don't think it's quite that simple, and I'll explain in a few slides if you know what I mean. Generally, what's happening with the autonomic nervous system is something like the hypothalamus, occasionally the other structures if they are tasked with, like, for instance, breathing, the pons and so forth, or some other types of rapid response pathways. They sense what's going on, either the hypothalamus or some other neuron. We send it in here. Now, this limbic system over here can influence. It says, is this a real threat or is this not a threat? It can influence the magnitude and the scope of the response. Perhaps you hear a book slam on a table and you jump for a second. You might jump, but you don't get up and run away. Okay. On the other hand, if you heard a gunshot, that might influence your limbic system enough to increase the magnitude of the response, and then you're running away, hiding under your desk, that sort of a thing. But it all interfaces with the effectors, either the neurons affecting direct neuronal control or indirectly through endocrine and or complex behaviors. Not a lot going on here, just showing you where these structures are that influence the autonomic nervous system. There are these discrete structures in here that we call these nuclei. They are called nuclei because it's a collection of neurons that are tasked with a very, very specific autonomic or endocrine function. Very, very specific homeostatic task. In the previous slide, I used the word oppositional. Another word for that is antagonistic. That is to say, they may amp up one division, like sympathetic, at the same time inhibit the other division. Or if you're looking at a given behavior, like for instance, heart rate, for instance, sympathetic would be excitatory in terms of increasing heart function, increasing rate, increasing strength of contraction. Where on the other hand, the parasympathetic and the vagus nerve, cranial nerve 10, okay, is involved in the parasympathetic side, and it dials down heart rate. If you're sitting in your chair and you're completely relaxed and your heart rate is somewhere between 60 and 70, that is because of the vagus nerve. The heart actually wants to beat closer to 70, 75, and the vagus nerve is actually dialing that down to slow that heart rate a little bit, an autonomic control. But if you get up and go somewhere, sympathetic will kick in, and it will start increasing not only the rate, but also the strength of the contraction. So heart is a great example of almost a pure antagonistic control. Though what's interesting is that sympathetic modulates not only rate, but also strength of contraction, whereas vagus really only modulates rate to a major degree. So if you've ever had a really, really good scare, I mean one where you just are shaking in your boots, you might notice that even though you're resting and your heart rate might slow down, it still takes you a while to sort of dial down the way you feel inside. Your blood pressure is still relatively elevated. Your heart is still kind of contracting. I had a situation where I thought I had heard somebody fall to their death, and thankfully they didn't. It was just their pack. And but nevertheless, it took me a good 20 minutes to kind of really settle down, even though I was resting and I didn't have a lot of metabolic need at that point. And that's in part because of the fact that we don't control everything exactly the same, and plus all those hormones are still circulating around like epinephrine. But fight or flight, rest and digest. It's a lovely, lovely way to recall this, and that's a good way to go from here. But I do want to give you some awareness that it's not always like that. That some of our more interesting behaviors and activities that we undergo are actually a combination therein. Have you ever found yourself sort of cycling through emotions? Sort of cycling through? Well, that's because the sympathetic and parasympathetic nervous systems work more like a pair of dancers. Yes, they are oppositional in their positions and often oppositional in the way that they're moving. One is moving forward while another is moving backward, but they work together. They work together, and there are activities where you actually have simultaneous sympathetic and parasympathetic stimuli or responses occurring simultaneously. Here are some good ones to refer to when we talk about sympathetic responses. For instance, pupil dilation. If you have, for instance, if you have a cat and you start playing with that cat with a cat toy, you can see their pupil, especially like one that looks like a little mouse tail or something underneath a rug. You can see their pupils dilate like crazy. So pupil dilation is a sympathetic side. However, dry mouth, and that's why whenever you see a major speaker at a lectern or something like that, they almost always have a glass of water there. That's because getting up in front of people is a very, very terrifying thing, even for people who do it all the time. And so you have a glass of water up there to help keep your mouth moist while you're speaking to however many people you are. And that's also indirectly because of mucus production decrease, heart rate, bronchodilation. If you ever had a cold or something like that and you've been working out, you might notice that if you start running or you start doing some sort of extended activity that you start opening up a little bit, and that's due to the bronchodilation aspect, and vice versa. So these are classic antagonistic: constriction for parasympathetic, increased salivation, increased mucus, bronchoconstriction. So that's why many of the asthma drugs that we have are actually what we call sympathetomimetics. That is to say, they mimic the sympathetic system to dilate the bronchioles, get that airway open up again, and then the side effects are things like racing heart or dry mouth or blurry vision. We call this pupil dilation, by the way, mydriasis, and pupil constriction meiosis. And you're going to see that, down the line, so I just decided to go ahead and include that. Just another chart showing the same thing, only just in a slightly different context. Parasympathetic involves increased digestive tone, decreased digestive tone. That can lead to either, like for instance, constipation or diarrhea in some cases. I don't have a ton to say in this because this is primarily anatomical, but the point I want to make is that the sympathetic pathways are what we would call divergent. What do I mean by that? When I mean divergent, it means it starts with common neurons and then it radiates out into a systemic response. Sympathetic responses tend to be much more systemic and much less targeted. and that is partly due to the fact that we have this sort of cross-talking and divergent anatomy in the neurons. And so we see thoracic neurons and we see lumbar neurons, that sort of thing. Parasympathetic, on the other hand, tends to be more targeted, tends to be specific to a region rather than systemic. Doesn't mean that it can't be systemic, but it tends to be more directly targeted. To have all the parasympathetic things occur all at once would be relatively unpleasant. It's a relatively unpleasant sensation. And so again, it's targeted. The big one in the parasympathetic that's kind of worth paying attention to is called the vagus nerve, cranial nerve 10. It is responsible for a big chunk of parasympathetic activity in the body. Please recall that earlier in this lecture I mentioned that all the autonomic system, whether it's sympathetic or parasympathetic and by extension adrenal, are a two-step system. With the sympathetic system, that two-step, the ganglia for those neurons is found in what is known as the sympathetic trunk. It's almost like two additional spinal cords in your body. There's one on one side, and then there's one not shown over here on the other side. The sympathetic trunks are where the neural cell bodies are that interface, the ganglia, if you will, between the pre-ganglionic neurons and the post-ganglionic neurons. So this is where that two-step pathway sort of occurs. Yeah, it's a little more complex, I know, but let's not worry too much about that. Mostly what you're seeing, for a lot of the big ones, you see a preganglionic neuron, the synapse is in the sympathetic trunk. And they're diverging. It's not only that they're interfacing with a downstream neuron. What will happen is if you send impulses down maybe some of these neurons to affect heart and lung, they crosstalk with each other. It's actually fascinating neurologic architecture. So you can send signals here and they may also carry over into other organ systems. It is what we call a divergent or diverging pathway system. And here we're seeing both the sympathetic and the parasympathetic. On the left, we have the sympathetic side. So here's that the sympathetic chain, if you will, the sympathetic trunk, the ganglion. And again, I don't want to get too fine with this. This is actually sort of a 3-neuron type system, but let's not worry too much about that. It's at least a 2-neuron system most of the time. Okay, at least a 2-neuron system. And so here are our preganglionic neurons, here are our postganglionic neurons. Look at the difference in the architecture with the parasympathetic. Here's vagus nerve, and look at all the branches it has. All right. You have facial nerves, oculomotor nerves for constriction of pupils, and then bladder and reproductive control below. But again, it's not as diverging as the sympathetic. This slide is a little bit of a carryover because in the past we wouldn't have had open notes, so I wanted to reinforce the idea of manifestations. And it's good to know the manifestations. It really is, because then you can identify, you can begin to narrow down what is the issue with the patient and come up with ways of intervening for those specific issues. If their heart rate's beating fast, why is it beating fast? Is it because they have increased metabolic need, or is it because they have anxiety? So we can narrow it down. Both have very different responses to deal with. So at least 2 neurons in series. I know that sympathetic's a little more complicated than that, but we're going to just treat it as 2. And I like this figure. It's showing a neuron as an arrow. So we have a single motor neuron going out to the effector tissue. We have a preganglionic neuron in the sympathetic, postganglionic neuron, preganglionic neuron, postganglionic. One more point to help you identify the difference. 1 neuron, but with the sympathetic, the preganglionic neuron is relatively short. It's relatively short. That's because that sympathetic trunk, or even the celiac ganglion, is relatively close, only a few centimeters away from the spinal cord. The Postganglionic neurons are relatively longer. With the parasympathetic, it's the opposite. The preganglionic neurons are relatively long, and the postganglionic neurons are relatively short. So you can identify these things not only by looking at them in a cadaver or on a figure, you can also identify them in abstract sense. You might recall from chapter 8 that neurons use any number of different kinds of neurotransmitters. Usually a neuron is dedicated to one form of neurotransmitter, but we do reuse transmitters across the body for different pathways and different responses. And that's one of the major causes of side effects and side manifestations, untoward outcomes in patients, is because the human body uses signals in different ways, the same signal in different ways. Well, in the autonomic nervous system, we have a subset of neurotransmitters that are used quite often. The first one is acetylcholine, and synapses and neurons that are involved in acetylcholine we call cholinergic, and they serve a huge role in the autonomic nervous system, but they also are very, very prevalent in the cerebral neurons. So again, side effects. In any case, cholinergic neurons utilize or are receptive to acetylcholine. There are two major flavors of receptors, and this is very, very important because these are going to be in different areas of the autonomic nervous system. We have on the left, we have what are called the nicotinic receptors. Nicotinic, sometimes we'll say nicotinic cholinergic receptor. So this is a subset of the cholinergic receptors, and it is also sensitive to nicotine. That's why it's called the nicotinic cholinergic receptor. It's receptive to both nicotine and acetylcholine. Then over here, in the other set of receptors is not only sensitive to acetylcholine, but it's sensitive to a compound that was originally identified in a type of mushroom that's mildly hallucinogenic, and that compound is called muscarine. And so these receptors are muscarinic cholinergic receptors. Then there's a third set of receptors that are sensitive to epinephrine and or norepinephrine. We call these adrenergic receptors because another name for epinephrine is adrenaline. Another name for norepinephrine is noradrenaline. So if you were to go to the UK or any of the Commonwealth nations that subscribe to the UK identity, they would say adrenaline, noradrenaline. But in these cases, these receptors have varying sensitivities to epinephrine and norepinephrine, and they are located in different areas. You'll hear about beta blockers or alpha agonists and that sort of a thing. We're not going to go there in this lecture. Nevertheless, you should have an awareness that this is coming, especially if you're going to be involved in prescribing authority type situations. What we're going to start doing now is move through the various neural pathways and talk about the neurotransmitters and show you the commonalities and ways to get a hold of this so that it's easier to remember. First of all, we have the somatic motor neuron here, and I put like a skeletal muscle here. I didn't put any stripes in it, but that's what that's supposed to be. And then we have our parasympathetic pathway, which is a long preganglionic neuron and a relatively short postganglionic neuron. And then here's our sympathetic, relatively short preganglionic. Then we have a long postganglionic. And the first thing to notice is that here, the first synapse, so this is the first synapse in that circuit. Right here, the first synapse in this circuit. And right here, the first synapse in this circuit, all are acetylcholine. They are all cholinergic synapses, and they all contain nicotinic cholinergic receptors. So the receptors on this muscle are nicotinic receptors. The receptors on this postganglionic neuron are nicotinic for parasympathetic, and the postganglionic neuron for sympathetic is also nicotinic. So that's one thing they all have in common, is that the first synapse, and this is true even in the adrenal gland, In the adrenal gland, you have a neuron going to the adrenal cortex, actually the adrenal medulla, my fault. And in the adrenal medulla, that's also a cholinergic situation with nicotinic receptors. And that's what explains the complex pharmacology of nicotine. It can have all kinds of effects on blood vessels, on the heart, on insulin and lipid metabolism. Certainly there are side effects that are not due to nicotine, but nevertheless it has lots and lots of functions, and that is probably the reason why is that nicotinic receptors are found all over the place. They are almost the entirety of the autonomic system. Certainly the neurons going to the skeletal muscles all have nicotinic receptors. They're not all the identical receptors. They may have varying sensitivity to nicotine, but nevertheless they are nicotinic. This figure right here is a summary of all of the different neurotransmitters and receptors for these two types of neuronal circuits. We've already talked about acetylcholine, so let's show you where those are. So there's the acetylcholine molecule down here. We have acetylcholine, so these are cholinergic synapses. So acetylcholine at the first synapse in all the circuits. And in the parasympathetic pathway, that synapse is also cholinergic. Now, if we look at the receptors for acetylcholine, the upstream receptors, these two right here, are nicotinic receptors. That is to say, they are sensitive to acetylcholine and nicotine to varying degrees depending on which tissue you're talking about. In the parasympathetic, the downstream receptor, the one there at the target tissue contains muscarinic receptors. That is to say, they're sensitive to acetylcholine and they are sensitive to muscarine, which is why muscarine is a parasympathetomimetic. That is to say, when you take muscarine, you'll have things like salivation, increased gland secretion, increased digestion. Occasionally diarrhea is a side effect of this. Certainly blurry vision. Blurry vision can go both ways. You can have sympathetic and parasympathetic issues, and both result in blurry vision. But moving on, we also have the sympathetic target tissue, which contains an adrenergic receptor that is sensitive to norepinephrine. Don't get that confused. Epinephrine does in fact work on many of these target tissues as well. It depends on which adrenergic receptor to the degree that epinephrine works. Epinephrine is sort of an example of that divergence that I was talking about earlier. We may have targeted neuronal control in a sympathetic way using norepinephrine, and then epinephrine, which is in the adrenal side of the equation, the adrenal gland, may come in as a hormone and augment norepinephrine. This is the summary picture from your book, which is okay. The reason I don't like it is that it should show a shorter preganglionic neuron for the sympathetic pathway and a longer preganglionic neuron for the parasympathetic, but it's showing the same information as before: cholinergic, cholinergic, adrenergic, nicotinic, muscarinic, adrenergic receptor. That downstream synapse between the neuron and the target tissue is often called the neuroeffector junction. In the case of the somatic one, it's the motor end plate is what they call it, but it's still a neuroeffector junction because you have a neuron and you have an effector tissue. That's the synapse between that postganglionic neuron. In this case, they're talking about specifically autonomic neurons, and the target cells, which can be smooth muscle, can be cardiac muscle, can be all those other tissues that we mentioned. These tissues do have varying functions, and so there is often a need for a varying type of neural structure. And we call these neural structures, in some cases, varicosities. And instead of having a single sort of, I don't know if dumbbell is the right word, shaped plate at the end, they often have a series of swollen areas that contain granules of, vesicles of neurotransmitter. So it allows them to stimulate any number of cells. And I'll have a picture for you here in just a second. But those neurotransmitters, just like anywhere else, diffuse across and bind to receptors in those varicosities. And here's what we're talking about with the varicosities. See, so you have these little bulbs, and those bulbs contain vesicles of neurotransmitter waiting to be released. Mitochondria there to make ATP to facilitate that exocytosis. But you can see the varicosities here. It's the same story as if it was just one presynaptic terminal, but it's just that it's a little bit of an anatomical difference, if you will. And I'm going to reinforce the idea that termination is critical. That termination, the ending of signaling, is critical. And we do that by modulating, by changing the concentration of neurotransmitter. So we can reduce it and reduce the effect or increase it. So diffusion, remember that we have three general ways of dealing with this: reuptake, enzymatic inactivation, and diffusion. So diffusion, it can be metabolized, so enzymatic inactivation. I'm generalizing here a little bit, and when I say that it's in the extracellular fluid, sometimes these things are brought in and then metabolized. I'll show you an example of that. Or they're a reuptake, but it's still the same story as before. It's just maybe a slight shade with just a little more detail there. For instance, monoamine oxidase, which is a target for certain antidepressants. Monoamine oxidase was a major antidepressant target for years and years and years. It's not quite as important now as it used to be. But in any case, what happens is when we take in, for instance, norepinephrine, it's brought in by reuptake. It is also enzymatically inactivated by monoamine oxidase. And so MAO inhibitors, what they do is they end up leading to an increase in norepinephrine in the synapse. Because when you increase it intracellularly, then it slows down the uptake. And so there's more norepinephrine extracellularly. But it's the same story. That's the key. That's why I want you to learn this so well. It's all, we can understand putting the neurotransmitter in here, but getting that neurotransmitter out and how we deal with it becomes a major issue for especially neuropharmacology and how we modulate certain types of emotional and neurocognitive disorders. So let's just look at the parallelisms of mechanism here. There's a lot of the same thing going on, even though it looks different than what we've seen. It is still the same story. Action potential causes a voltage shift. Voltage-gated calcium channel causes exocytosis of norepinephrine into the synaptic space, where it can either bind a receptor if there's enough of it. Remember, loss mass action here. It can diffuse away, which it often will do. We don't have enzymatic inactivation in the space here shown, but there is going to be some. But that norepinephrine can be actively transported back into the cell where it can either be repackaged into more norepinephrine or it can be degraded by monoamine oxidase. But even though it looks different, it's the same story, just as we saw in chapter 8. Just the same story, just a slightly different variation from acetylcholine. Main difference with acetylcholine is that instead of monoamine oxidase breaking it down up in here, acetylcholinesterase breaks it down out here. But it's still brought in. This one's secondary active transport. And then we repackage acetylcholine. With norepinephrine, it's brought in first, and it's either degraded by monoamine oxidase from the mitochondria, or it's repackaged into norepinephrine. I want to show you this because why we learn what we learn now sets you up to make it easier for you to learn stuff down the line. Like for instance, the adrenergic receptors, there's a whole set of, several different subforms. Some are reactive mostly to norepinephrine, some are equally reactive, and then some are exclusively reactive to epinephrine. And where they are in the tissue leads to how we can track down what the side effects are, or we can target tissue specifically. Like maybe we want to just do something that treats someone's heart but doesn't cause other side effects. We might go for a beta 1. That's where this sort of goes. I'm not going to test you on tissue distribution and isoform at this point, but you certainly the muscarinic and the nicotinic and adrenergic receptors are fair game for exam purposes in this class. To tie back what we have talked about in the past when it comes to membrane receptors, if we look at adrenergic receptors, what you'll notice is that they affect a lot of cyclic AMP. They are receptor enzymes. Cyclic AMP. Now one decreases it, one increases it. Phospholipase, that's just a phospholipid cleaving enzyme. In any case, you can see that they're all dealing on the receptor enzyme level. They're all receptor enzymes, the adrenergics. For the muscarinics, again, we have lots and lots of isoforms for those, and they're tissue restricted and so forth. They're all G protein coupled receptors. They all do that same pathway. So again, that's why I'm showing you the reasons why we learn these things in the first place, so that we can have these conversations and see similarities across receptors down the line. And so there are the muscarinics. They are G protein coupled receptors. We're going to finish up with the last couple of slides with our final autonomic pathway, which is the adrenal medulla, the middle of the adrenal gland. Like the pituitary, the adrenal gland is actually composed of two different kinds of tissue. The outer tissue is synthesizing steroids. The inner tissue, which is believed to originate from the same precursors as neurons, that's why we call it neuroendocrine tissue, synthesizes catecholamines. That is to say, primarily epinephrine and norepinephrine. So let's look at this pathway. These cells are like the second neuron in the neuronal pathways that we've talked about. So here's the first neuron. Here is the preganglionic sympathetic neuron. And just like before, we have a cholinergic synapse in the adrenal medulla. So we have acetylcholine and the receptors on those neuroendocrine cells is nicotinic. So cholinergic, nicotinic. This time, though, our second neuron is not a neuron at all. It's hormone, in this case, epinephrine. So instead of a neural pathway, we have a hormonal one. So our second signal is epinephrine, and you can guess what that receptor is going to be. It's going to be an adrenergic receptor. Remember that the other word for epinephrine is adrenaline. So it's going to be an adrenergic receptor of some type. So let's finally put this all together. Parasympathetic, we have cholinergic, nicotinic, cholinergic, muscarinic. And again, you can notice the targets here. Sympathetic, cholinergic, nicotinic, norepinephrine, I'll say adrenergic there, norepinephrine, adrenergic receptor. For the adrenal medulla, cholinergic, nicotinic, epinephrine, adrenergic. And this is a really nice slide, by the way. Good reference for you. What are the autonomic targets? Smooth and cardiac muscle, glands, endocrine and exocrine. For instance, if you're hungry and I say, and let's say you're a chocoholic and I say chocolate, well, what will happen is you might start salivating, and that's an exocrine gland in your oral cavity. Then we also have adipose tissue that can do this, and there's a wide diversity within those three subsets of targets. Sympathetic, remember that it's a diverging situation. We have that sympathetic ganglia, that sympathetic trunk that crosstalks. And with the sympathetic, we also have this widely diverging adrenergic pathway using epinephrine. And that concludes what I think is one of the most interesting and applied chapters that we have this semester. Yeah, there's a fair amount going on in terms of detail here. The key is start simple, work through the rules that bind these things together, and then the detail will come along as you gain experience. So work on these basic neuronal circuits, work on the structures that we talked about, and you should be in good shape. Thank you for your time, and I will talk to you again when we start talking about sensors, starting with somatic senses.